Right now, as you're watching this, your body is forming microscopic cancers. Not maybe, not possibly, it's happening right now. Research shows that by age 40 to 50, about 40% of women have microscopic breast cancers. By age 50 to 60, roughly 50% of men have microscopic prostate cancer. And by age 70, nearly everyone has microscopic thyroid cancer. But here's what's remarkable. Most of us will never know they're there. These tiny clusters of abnormal cells, no bigger than the tip of a ballpoint pen, will remain harmless for our entire lives. Our bodies have built-in defense systems that keep them in check, prevent them from growing, and in many cases eliminate them entirely. What makes the difference between a microscopic cancer that stays dormant and one that becomes dangerous? Blood supply. The moment a tumor hijacks your body's blood vessel growth system and gets fed, it can grow 16,000 times its size in just 2 weeks. 16,000 times in 14 days. Today, I'm going to show you the foods that activate your body's natural defense systems. Foods that enhance DNA repair mechanisms damaged by over 19,000 hits per day. Foods that can actually cut off the blood supply tumors need to grow. But before we go any further, I need to be crystal clear about something. What I'm sharing with you today is about prevention and support, not cure. If you're currently dealing with cancer, everything we discuss should be coordinated with your oncology team. These foods work alongside conventional treatment, not instead of it. This is critically important. Now, let's talk about what's actually happening inside your body right now. Your DNA is under constant attack. Every single day, each cell in your body experiences approximately 19,000 DNA lesions. 19,000! That's from normal metabolism, from breathing oxygen, from sunlight, from chemicals in your environment. Your DNA is constantly being damaged, and if left unrepaired, these lesions turn into mutations. Accumulate enough mutations in the wrong genes, and you get cancer. But your body's not defenseless. You have 5 major DNA repair systems working on the clock. Base excision repair fixes damaged individual DNA bases. Nucleotide excision repair removes bulky damage caused by UV light and chemicals. Mismatch repair corrects errors that happen during DNA copying. And 2 systems, homologous recombination and non-homologous end joining, fix the most dangerous damage of all, double-strand breaks where your DNA physically snaps in two. These repair systems need specific nutrients to function. Think of them like a construction crew that needs the right tools and materials. Without folate, without B12, without zinc, selenium, and other micronutrients, your DNA repair machinery can't do its job effectively. Here's what happens when you don't get enough folate. Your cells start misincorporating uracil into DNA instead of thymine. This creates instability, strand breaks, and increases your cancer risk. A 2024 study found that folate deficiency causes DNA damage comparable to radiation. Comparable to radiation. But when you provide your body with the right nutrients, remarkable things happen. And some foods don't just supply basic building blocks. They actively upregulate your DNA repair genes, making your repair systems work more efficiently. Let me show you the most powerful foods for DNA repair, starting with the ones that have the most impressive research behind them. Berries, specifically red raspberries, blackberries, and blueberries. These aren't just healthy snacks, they're DNA repair activators with quantified effects. A landmark 2008 study fed mice either red raspberry or isolated ellagic acid, one of the key compounds in berries. What they found was extraordinary. Red raspberry reduced endogenous DNA adducts by 59%. That's a 59% reduction in DNA damage markers, with statistical significance at p less than 0.001. Ellagic acid alone achieved a 48% reduction. But here's where it gets even more interesting. When researchers looked at gene expression, they found that berry consumption caused a 3 to 8-fold overexpression of critical DNA repair genes. Genes like XPA, ERCC5, and DNL3. These are components of nucleotide excision repair and base excision repair pathways. The berries didn't just reduce damage, they ramped up the repair machinery itself. Human studies back this up. A 2024 study found that people consuming mixed berries for just 4 to 6 weeks showed 15% less DNA damage compared to controls. Another study from 2020 reported that 300 grams of berries daily reduced oxidative DNA lesions by approximately 20% after 6 weeks. 20%! Now, not all berries are equal in terms of concentration. Blueberries, for example, are particularly rich in anthocyanins. These are the blue pigments that also happen to be powerful immune system activators. Research shows that eating about 1.5 cups of blueberries daily can elevate your T cells by 88%. T cells are the super soldiers of your immune system that identify and destroy cancer cells before they become dangerous. And here's what makes this even more compelling. Anthocyanins simultaneously lower inflammation while boosting immunity. Most people think you have to choose between a strong immune response and low inflammation, but blueberries give you both. The practical recommendation: 2 cups of mixed berries daily. Fresh or frozen doesn't matter. The compounds are stable. You can rotate between raspberries, blackberries, blueberries, and strawberries to get a variety of beneficial compounds. Now, let's talk about another food with exceptional DNA protection data: kiwi. Kiwi fruit contains high levels of vitamin C, which acts as a powerful antioxidant protecting your DNA from oxidative damage. But what's remarkable about kiwi is the dose-response relationship researchers have documented. Studies show that eating just 1 kiwi a day protects DNA against damage by about 60%. Eat 3 kiwis, and your body can rebuild damaged DNA by approximately 60%. These are human studies with measurable outcomes. But kiwi does something else that's equally important. It's packed with fiber, making it essentially a prebiotic food that feeds your gut microbiome. Research from Singapore demonstrated that eating even 1 kiwi overnight begins changing your gut microbiome composition, favoring healthier bacterial species. Why does this matter for cancer? Because your gut microbiome is intimately connected to your immune system's ability to fight cancer. A groundbreaking 2022 study looked at 200 cancer patients receiving immunotherapy. The researchers found that the only difference between patients who responded well to treatment and those who didn't was a single bacteria in their gut: Akkermansia mucinifila. Patients with adequate levels of Akkermansia had dramatically better responses to immunotherapy. And you can't take Akkermansia as a probiotic. You have to grow it yourself by feeding your gut microbiome the right fibers. Foods like kiwi, pomegranate, cranberry, and whole grains create the environment Akkermansia needs to thrive. Now let's talk about cruciferous vegetables: broccoli, Brussels sprouts, cauliflower, kale, cabbage. These vegetables contain compounds called glucosinolates that break down into sulforaphane and indole 3-carbinol when you chew them. Sulforaphane activates your body's antioxidant defense systems and enhances DNA repair capacity. A comprehensive review found that consuming about 8.8 ounces of steamed broccoli daily increased DNA protection, particularly in smokers who have higher baseline DNA damage. But here's a critical detail most people don't know. The stems of broccoli contain twice as much sulforaphane as the florets. Don't throw the stems away. Chop them finely, blend them into soup, or saute them. You're doubling your sulforaphane intake. And if you really want to maximize the benefits, eat broccoli sprouts. These are 3 to 4 day old baby broccoli plants. Broccoli sprouts contain 50 to 100 times more sulforaphane than mature broccoli. A study gave healthy young adults a broccoli sprout shake before they received the flu vaccine. The result? Their immune response was amplified 22-fold compared to the control group. 22 times stronger immune activation from a simple broccoli sprout shake. Multiple studies have linked cruciferous vegetable consumption with reduced cancer risk. The health professionals' follow-up study found that men eating cruciferous vegetables at least 5 times per week had a 51% lower risk of bladder cancer. The nurses' health study showed a 33% reduction in non-Hodgkin's lymphoma risk with regular cruciferous vegetable consumption. These aren't small effects. These are substantial risk reductions from common vegetables available at any grocery store. Now, I need to address something important about DNA repair and supplements. You might be thinking, if these nutrients are so important, can't I just take a multivitamin and get the same benefits? No. And the research on this is actually quite alarming. A Cochrane systematic review analyzed 20 randomized clinical trials involving over 211,000 participants. They looked at antioxidant supplements, including beta-carotene, vitamin A, vitamin C, and vitamin E for cancer prevention. The result? No convincing preventative effect. None. But it gets worse. The mortality data showed that 14% of people taking antioxidant supplements died during the study period, compared to 11.2% taking placebo. Antioxidant supplements were associated with increased mortality. The only exception was selenium, which showed potential preventative effects, but even that requires further confirmation. Why don't supplements work when the whole foods clearly do? Because food is a complex matrix. When you eat a blueberry, you're not just getting anthocyanins. You're getting hundreds of different bioactive compounds, fiber, water, and other nutrients that work synergistically. The whole is greater than the sum of its parts. Your body recognizes food. It doesn't recognize isolated compounds compounds in pill form the same way. Bioavailability is different. Metabolism is different. Effects are different. This is why I keep emphasizing whole foods throughout this video. The research supports whole food consumption, not supplementation. Now, let's shift gears and talk about how certain foods can actually attack cancer cells that have already formed. Remember what I said at the beginning? A microscopic tumor without blood vessels cannot grow larger than the tip of a ballpoint pen. It's stuck. It doesn't have enough oxygen or nutrients to expand. But the moment blood vessels reach that tumor and start feeding it, explosive growth begins. A tumor that's the size of a pinhead can grow 16,000 times in just 2 weeks once it gets a blood supply. This process is called angiogenesis, the formation of new blood vessels. And cancer cells hijack this system by secreting growth factors that act like fertilizer, stimulating blood vessel growth directly toward the tumor. Your body normally tightly regulates angiogenesis. Blood vessels grow when needed for healing, then growth stops. But tumors bypass these controls and force continuous blood vessel formation. Here's what's fascinating. More than 100 foods contain natural compounds that can inhibit angiogenesis. They cut off the blood supply feeding tumors. Let's start with green tea. Green tea contains polyphenols, particularly epigallocatechin 3 gallate, or EGCG. When researchers tested EGCG using the same laboratory methods used to develop anti-cancer drugs, they found it was remarkably effective at stopping blood vessel growth toward tumors. EGCG inhibits topoisomerase 1, an enzyme involved in DNA replication that rapidly dividing endothelial cells need to form new blood vessels. It also inhibits arachidonic acid metabolism by 30 to 75% at concentrations of 30 micrograms per milliliter. Arachidonic acid metabolites promote inflammation and angiogenesis, so blocking this pathway has anti-cancer effects. Multiple animal studies have shown that green tea catechins suppress VEGF, vascular endothelial growth factor, the primary signal tumor cells use to trigger blood vessel growth. Now, I need to mention an important limitation here. EGCG has relatively low bioavailability. After you drink green tea, less than 2% of the EGCG actually makes it into your bloodstream in active form. Your liver and intestines heavily metabolize it. Does this mean green tea doesn't work? No. Population studies consistently show that regular green tea consumers have lower cancer rates, but it does mean you need to consume it regularly, not just occasionally, to maintain beneficial blood levels. 3 to 5 cups of quality green tea daily appears optimal based on population studies. Let's talk about tomatoes. Tomatoes contain lycopene, a carotenoid that gives them their red color. Lycopene has anti-angiogenic properties and has been studied extensively in relation to cancer prevention. A 2025 meta-analysis just published analyzed 119 prospective studies, including 108,574 cancer cases. High lycopene intake compared to low intake was associated with a 5% reduction in cancer incidence. The pooled relative risk was 0.95 with a 95% confidence interval of 0.92 to 0.98. More importantly, when researchers looked at blood lycopene levels instead of dietary intake, they found an 11% reduction in cancer mortality with elevated serum lycopene. A Harvard study specifically looked at tomato products and prostate cancer. Men consuming tomato products regularly had 40 to 50% lower rates of fatal prostate cancer. And when researchers examined tumor tissue, they found that tomatoes reduced angiogenesis markers within the tumors themselves. Here's a practical tip. Lycopene bioavailability increases dramatically when tomatoes are cooked and consumed with fat. Fresh tomatoes give you about 4% lycopene absorption. Cooked tomatoes with olive oil can increase absorption to 15 to 20%. So tomato sauce, tomato paste, and cooked tomatoes are actually more beneficial than raw ones for this specific compound. Aim for 2 to 3 servings per week of cooked tomato products. Now let's talk about resveratrol, found in red grapes, red wine, and some berries. Resveratrol is one of the most researched polyphenols for anti-angiogenic effects. In melanoma models, resveratrol combined with 5-fluorouracil significantly reduced VEGF production and inhibited blood vessel formation toward tumors. In breast cancer xenograft models, resveratrol suppressed angiogenesis through inhibiting NF-kappa B regulated VEGF production. In human terms, resveratrol interferes with the signals tumor cells use to recruit blood vessels. The challenge with resveratrol is similar to EGCG. Bioavailability is low. You'd need to consume unrealistic amounts of red wine to reach the concentrations used in animal studies. But Concord grape juice, red grapes, and certain berries provide resveratrol along along with dozens of other polyphenols that work synergistically. The research suggests that the combination of compounds in whole foods produces effects that isolated resveratrol supplements can't match. Other anti-angiogenic foods with research backing include soy products containing genistein. Genistein has been shown to downregulate VEGF and matrix metalloproteinases in multiple cancer models. And contrary to internet myths, soy does not increase breast cancer risk. Phytoestrogens in soy look nothing like human estrogens chemically. They actually block estrogen receptors and have anti-cancer properties. Multiple studies in breast cancer patients show that soy consumption is associated with better outcomes, not worse. Berries, again, beyond their DNA repair properties, berries suppress VEGF expression independent of their antioxidant activity. This is a separate mechanism. Quercetin, found in onions and apples, inhibits endothelial cell proliferation and suppresses MMP2, an enzyme that breaks down tissue barriers, allowing blood vessels to invade toward tumors. The practical takeaway here is dietary diversity. You want multiple anti-angiogenic compounds working through different mechanisms. A tomato sauce with garlic and onions, seasoned with herbs, and served with green tea hits multiple anti-angiogenic pathways simultaneously. Now, I want to address the idea of starving cancer through diet. This is where we need to get into some sophisticated biology. Cancer cells fundamentally differ from normal cells in how they produce energy. This is called the Warburg effect, discovered by Otto Warburg in the 1920s. Normal cells, when oxygen is available, use a process called oxidative phosphorylation in their mitochondria. This generates about 36 to 38 molecules of ATP per glucose molecule. It's highly efficient. Cancer cells, even when oxygen is abundant, preferentially use glycolysis. Glycolysis only produces 2 molecules of ATP per glucose. It's incredibly inefficient. So why would cancer cells do this? Because they're not optimizing for energy efficiency. They're optimizing for rapid growth. Glycolysis provides biosynthetic precursors for building new cellular components. Nucleotides for DNA, amino acids for proteins, lipids for membranes. Cancer cells are building mass as fast as possible. The consequence is that cancer cells consume glucose at rates 10 to 200 times higher than normal cells. They're glucose addicted. This creates a potential vulnerability. If you reduce glucose availability, can you selectively starve cancer cells? The answer is, it's more complicated than a simple yes or no answer. Several metabolic dietary interventions have been tested: ketogenic diets, fasting mimicking diets, and specific amino acid restrictions. Let me walk you through what the research actually shows. Ketogenic diets drastically reduce carbohydrate intake to less than 50 grams per day, often as low as 20 to 30 grams. This forces your body into ketosis, where it produces ketone bodies from fat to use as fuel instead of glucose. The theory is that cancer cells can use glucose but struggle to use ketones efficiently. So a ketogenic diet might selectively starve tumors while normal cells adapt to burning ketones. A 2024 study tested this in glioblastoma, one of the most aggressive brain cancers. 18 patients followed a ketogenic diet during standard treatment. The study found that patients who maintained ketosis for at least 50% of days had longer survival compared to historical controls. Median progression-free survival was 12.9 months, and overall survival was 29.4 months. These are encouraging numbers for glioblastoma, which typically has a median survival of 12 to 15 months with standard treatment alone. However, this was a small phase 1 trial without a randomized control group, so we can't draw definitive conclusions yet. Animal studies show more dramatic results. A meta-analysis of 17 animal studies found that ketogenic diets reduced tumor weight with a standardized mean difference of -2.45, reduced tumor volume, and prolonged survival across various cancer types. But here's a critical limitation. Some tumors actually adapted to use ketones. In certain melanoma models, ketogenic diets accelerated tumor growth rather than slowing it. Tumor metabolic flexibility varies dramatically by cancer type and genetic profile. The human clinical evidence for ketogenic diets in cancer remains preliminary. Most studies are small, uncontrolled, or measure feasibility rather than efficacy. A 2023 review concluded that while ketogenic diets can reduce blood glucose in cancer patients, they have not shown consistent improvements in other clinical endpoints across heterogeneous trials. Fasting mimicking diets are different. Instead of chronic ketosis, FMD involves periodic cycles of very low-calorie intake designed to mimic the metabolic effects of fasting while providing minimal nutrition. A 2022 study published in Cancer Discovery enrolled 101 cancer patients who underwent fasting mimicking diet cycles alongside their standard treatment. The FMD was safe and feasible. It produced consistent decreases in blood glucose, serum insulin, and IGF-1. IGF-1, insulin-like growth factor 1, is a potent growth signal. Lower IGF-1 means less proliferative drive. After 3 FMD cycles, IGF-1 decreased
increased by approximately 21.7 nanograms per milliliter with statistical significance at p equals 0.0017. The study also found that FMD reshaped systemic immunity, reducing immunosuppressive myeloid cells and regulatory T cells while enhancing tumor-infiltrating cytotoxic T cells. In other words, it made the immune system better at attacking cancer. But again, this study measured metabolic and immune biomarkers, not survival outcomes. The authors explicitly called for phase 2 and phase 3 trials to determine if these metabolic and immune changes translate into better cancer control. Animal studies are more definitive. Multiple mouse studies have shown that fasting or FMD cycles delay tumor growth, enhance chemotherapy response, and reduce treatment toxicity. In some neuroblastoma models, fasting plus chemotherapy produced long-term cancer-free survival when either treatment alone did not. The mechanism appears to be differential stress resistance. Short-term fasting shifts normal cells into a protective mode by lowering IGF-1 and activating stress resistance pathways. But cancer cells, driven by oncogenes, cannot enter this protective state. They remain vulnerable. So fasting simultaneously protects normal tissues and sensitizes tumors to chemotherapy. This is a remarkable biological phenomenon with strong preclinical support and emerging clinical data. Now, here's a critical caveat I need to emphasize. Fasting or severe caloric restriction is not appropriate for everyone, especially not for cancer patients. Many cancer patients are at risk for malnutrition, muscle wasting, and cachexia. Unmonitored fasting can cause serious harm in these populations. All of the clinical trials using fasting or FMD in cancer patients carefully screen participants, exclude those at risk at nutrition, and closely monitor body composition throughout. These interventions are done under medical supervision. If you're dealing with cancer, do not attempt fasting or FMD without coordination with your oncology team and a registered dietitian who specializes in oncology nutrition. This is not something to try on your own. The other metabolic vulnerability researchers are exploring is amino acid dependency. Many tumors are addicted to specific amino acids and cannot grow when those amino acids are restricted. Methionine restriction has shown the most promise. Many cancer cells cannot synthesize methionine efficiently and depend on dietary methionine. Restricting methionine slows tumor proliferation in multiple preclinical models. Small pilot clinical trials combining methionine restriction with chemotherapy have reported feasibility and occasional tumor responses. But here's where it gets complicated. A 2023 study found that methionine restriction can impair anti-tumor immunity through effects on gut microbiota and hydrogen sulfide production. In some contexts, methionine restriction sensitizes tumors to therapy. In other contexts, it weakens the immune response. The net effect depends on tumor type, immune status, and other factors we don't fully understand yet. This is why metabolic interventions remain investigational. The biology is complex, context matters, and what works in mice doesn't always translate directly to humans. The practical takeaway? Metabolic interventions show genuine promise as adjuncts to conventional cancer treatment, but they require careful implementation, patient selection, and medical oversight. They are not do-it-yourself cancer treatments. For prevention, the story is clearer. What you can do starting today. The evidence for cancer prevention is strong. A comprehensive analysis found that approximately 30 to 40% of cancer cases are preventable through diet, physical activity, and maintaining healthy body weight. 30 to 40%. That's not a small number. That's potentially millions of cancer cases that never need to happen. Here's what the research supports. Consume at least 5 servings of fruits and vegetables daily. A study of 2,381 participants found that those consuming 5 or more servings had 86% lower cancer risk compared to those who didn't meet this threshold. Every additional serving beyond 5 provided an 11% further risk reduction. These aren't theoretical benefits. These are observed outcomes in real populations. Emphasize variety and color. Different pigments represent different classes of protective compounds. Eat deeply colored vegetables and fruits, dark leafy greens, red and orange vegetables, blue and purple berries. Rotate through them. Include berries daily. Based on the DNA repair studies, aim for about 2 cups. Fresh or frozen, doesn't matter. The beneficial compounds are stable through freezing. Consume cooked tomatoes regularly. 2 to 3 times per week provides consistent lycopene intake. Use olive oil when cooking them to enhance absorption. Drink green tea. 3 to 5 cups daily appears optimal based on population studies. If you're sensitive to caffeine, drink it earlier in the day. Support your gut microbiome with prebiotic and fermented foods. Kiwi, Pomegranate, Concord grapes, whole grains provide prebiotic fiber. Fermented foods like yogurt, kefir, sauerkraut, and kimchi support beneficial bacteria. Follow a Mediterranean-style dietary pattern. This isn't a rigid diet plan. It's a pattern. Abundant vegetables, fruits, legumes, whole grains, olive oil, fish, moderate amounts of dairy and poultry, limited red meat. A study found that high adherence to Mediterranean diet was associated with 29% reduction in gastric cancer risk. Ensure adequate B vitamin intake, particularly folate and B12. Leafy greens, legumes, fortified grains provide folate. B12 comes from animal products or fortified plant-based foods. These vitamins are essential for DNA synthesis and methylation. Get sufficient zinc and selenium. Zinc from oysters, red meat, nuts, and seeds. Selenium from Brazil nuts, seafood, and selenium-rich grains. Just 2 Brazil nuts per day provide adequate selenium. More than that can cause toxicity, so don't overdo it. Limit red and processed meat consumption. Strong evidence links processed meat to increased colorectal cancer risk. The World Health Organization classifies processed meat as carcinogenic to humans. Limit red meat to no more than 12 to 18 ounces of cooked meat per week. Choose fish, poultry, or plant-based proteins most of the time. Maintain a healthy body weight. Excess body fat is linked to increased risk of at least 12 different cancer types. Even modest weight loss in overweight individuals reduces cancer risk. Limit or avoid alcohol. Alcohol is a known cause of several cancers, including breast, colorectal, liver, and esophageal cancer. For cancer prevention, less is better. If you choose to drink, limit to 1 drink per day for women, 2 for men. Now, here's something important. These recommendations aren't about deprivation. This isn't about what you can't have. It's about what you can add to create an eating pattern that activates your body's natural defenses. You don't have to be perfect. You don't have to follow every recommendation every day. The goal is consistency over time, a dietary pattern that's sustainable for years and decades. And here's something I want you to understand about the psychology of this. When you spend time thinking about what to add to your diet that protects you, you naturally spend less time thinking about the foods that increase risk. When you fill your meals with protective foods, there's less room for the problematic ones. This isn't about willpower or restriction. It's about empowerment and addition. Everything we've discussed today sits within a larger cancer biology that's worth understanding. Cancer isn't one disease. It's hundreds of diseases with different genetic drivers, different metabolic profiles, and different vulnerabilities. What works for one cancer type might not work for another. This is why we can't make blanket statements like this food cures cancer. But what we can say, based on solid evidence, is that dietary patterns rich in the foods we've discussed consistently reduce overall cancer risk across population. The mechanisms are diverse: DNA repair enhancement, angiogenesis inhibition, immune system support, reduction of inflammation, favorable changes to gut microbiome composition. These mechanisms don't just affect cancer. They affect your overall health. Better DNA repair means healthier aging. Reduced inflammation means lower risk of cardiovascular disease, diabetes, and neurodegenerative disease. A healthy gut microbiome affects everything from mood to immune function to metabolic health. The foods that reduce cancer risk are the same foods that promote longevity and health span. You're not just preventing cancer. You're optimizing your body's defense systems across the board. And here's what I find most empowering about this. You have control over these factors. You can't control your genetics. You can't eliminate environmental exposures, but you can control what you put on your plate 3 times a day. That daily choice, repeated consistently over time, has measurable impact on your cancer risk. A 30 to 40% reduction in cancer risk is achievable through dietary modification. That's based on rigorous epidemiological research, not speculation. Now, I want to address one more time the limitations and cautions. If you currently have cancer, everything in this video should be discussed with your oncology team before implementation. Some of these foods and compounds can interact with chemotherapy or other treatments. Timing matters. Context matters. Metabolic interventions like fasting or ketogenic diets require medical supervision in cancer patients. Do not attempt these on your own. Malnutrition and muscle wasting are real risks that can worsen outcomes. Supplements are not equivalent to whole foods. The research consistently shows that isolated nutrients in pill form don't provide the same benefits as whole food sources. Some supplements can even be harmful during cancer treatment. These dietary strategies are preventative and supportive. They work alongside conventional treatment, not instead of it. Don't delay or refuse evidence-based cancer treatment in favor of dietary approaches alone. That decision can be life-threatening. But with those cautions firmly in mind, the evidence is clear. Food is powerful medicine. Your body has sophisticated defense systems that can prevent most cancers from ever becoming dangerous. These systems need proper nutrition to function optimally. When you provide your body with the right compounds through whole foods, you enhance DNA repair capacity, support immune surveillance, inhibit angiogenesis, and create an internal environment that's hostile to tumor development. These aren't theoretical mechanisms. These are measurable biological effects with clinical correlates and population health. The foods we've discussed today, berries, cruciferous vegetables, kiwi, tomatoes, green tea, and a Mediterranean-style dietary pattern rich in diverse plant foods. These are accessible, affordable, and supported by decades of research. You can start implementing these strategies today. Add a cup of berries to your breakfast. Include broccoli or Brussels sprouts with dinner 3 times this week. Drink green tea in the afternoon instead of coffee. Cook a tomato-based sauce with garlic and onions. Small, consistent changes compound over time into significant risk reduction. Your body is working every moment to maintain your health, to repair damage, to eliminate abnormal cells before they become dangerous. Give your body the tools it needs to do this work effectively. That's the message I want you to take from this video. You're not powerless. You have agency, and the choices you make today affect your cancer risk tomorrow. If you found this information valuable, make sure you're subscribed because I'll be diving deeper into specific aspects of nutritional oncology and prevention strategies in upcoming videos. Drop a comment below with questions or topics you want me to cover next. And remember, this isn't about perfection. It's about consistent, sustainable choices that support your body's natural defenses. You've got this.